So, welcome everybody. This morning we had two sessions on insect vectors. And the first one will be chaired by Domenico Bosco from Università di Torino. And the second one will be chaired by Alberto Ferreres from Spain. And so I ask all the speakers in this today and tomorrow to come and stay on board so we can make space for everybody in the room. Thank you very much. So I let the floor to Domenico. I see somebody still coming, so maybe we wait 1 minute. Or
okay, so
yeah.
Okay, thank you, Giuseppe. We wait just a couple of minutes.
Yeah.
So. To reach the university, okay, but at least the speaker should.
Lo dici tu agli speaker di là o lo dico io adesso?
Ok, glielo dico io.
Come vuoi tu?
Ma dillo tu.
And for all the speakers, the colleague in the first row, she will show you the card, the yellow card, but turn to the other direction. Yeah. The yellow card means 3 minutes to the end. Then we have another card is red, 1 minute to the end. So this will help you to regulate your speech.
This is the most
and game over when you have finished your 25 minutes for the keynote speech and 15 minutes for the other speech.
I think, okay. So, good morning and welcome to the session on insect vector and epidemiology. We had plenty of very interesting contribution on different aspects of vector biology and their role in transmission. So, it's with great pleasure that I invite the first speaker to open this session, Joao Lopes from the University of São Paulo in Piracicaba, Brazil. As for the session of yesterday, the discussion, we leave the discussion for at the end of the presentation. So we have a final discussion for the first part of this session at more or less at 10:30. So we can start with the presentation.
Okay. Thanks, Domenico. Good morning, everyone. I thank you, EFSA, and other organizers of this meeting for the invitation. It's a great pleasure to be here and share some experiences on vectors of Xylella fastidiosa. In this talk, I will focus on diversity of vectors, but the main question is what make they important? What factors contribute to epidemiological importance of this species, of particular species? So, as you see, Xylella fastidiosa pathosystems are very complex. You have pathogen genetic variation. You have different subspecies, different sequence types. Even in the same crop, you may have different subspecies or sequence types and variation. You have also, in general, many host plants. You have several vector species. in many cases, in other cases not, but in particular situations you can have several species. And affecting all these disease elements, you have the abiotic factors that interfere with the occurrence of plants and vectors, and therefore having implications on epidemiology and on symptom expression of this disease. So in order to manage diseases caused by Xylella, you need to have a broad understanding of all these elements: host plants, vectors, the pathogen, abiotic factors influence. And the disease is very context dependent. If you change a particular vector species or you have a new host plant, you may change the epidemiology. There are many reports of this in the literature, so you need to understand that. In this talk, I'm going to talk about this diversity of vectors. And so, as I said, the question is, what vector species play a major role in disease progress? What are the key vectors? And also, what's the role of communities of sharpshooters in disease ecology? So, if you have many vectors, you can have different processes going on. And determining what vector species are and inoculum sources are relevant for pathogen spread. This is basic for understanding and the disease management strategy, and proposes a very rational method for controlling the disease. So vectors of Xylella are diverse. Because the bacterial is xylem limited, the vectors are groups of Auchenorrhyncha that are specialized in xylem feeding. So you have the sharpshooters as a major group. There are the leafhoppers in the subfamily Cicadelini. There are two tribes, Cicadelini and Proconiini, that represent 80% of the reported vector species. But also you have spirobugs, which in Europe are very important. So we have some families like Aphrophoridae, Clastopteridae, Cercopidae, which also contain vector species. And one report or two of transmission by cicadas, which are also xylem feeders. This is more or less the number of reported vectors per crop. So as you see, among the major affected crops, you have, in general, 4 or more species reported. And in some cases, like citrus and grape, you can have 14, 15 reported vector species. So many vectors in place to transmit the pathogen. And besides those species that are confirmed as vectors, you have many other species that are candidates, that are potential vectors, because they are also xylem feeders. They belong to these groups, and they are expected to transmit the pathogen, because there is a very low vector specificity. So if you have other species that have not been tested by transmission assays, it's very likely that they will transmit if you test them in sufficient numbers. So, for example, if you have a very low transmission rate, like 1%, sometimes it's required to test like 300, 400 insects in order to detect these very low transmission rates. So sometimes it's difficult and they are potential vectors. Epidemiological role is not well known for most of the vector and these potential vector species. So this is a big challenge. You need some kind of information to determine this. So citrus variegated chlorosis in Brazil and also the Pierce's disease in grape are examples of a complex situation in terms of vectors. You have many vector species, and it's important to understand the epidemiological role of particular species. So when the disease appeared in Brazil, the first step to search for vectors was to understand the community, to characterize the community of xylem-feeders. And several studies have been done with sampling methods. And since the beginning, there was a perception of a large number of genera of Cicadelline leafhoppers, such as Cicadelline, Proconiini, many genera, and also some Cercopodia, especially Cercopids, in citrus orchards. So around 29 genera. So the next step was to test all these candidates for transmission from citrus to citrus. And experiments were designed to test species from these candidate groups, such as Cicadelines and also Cercopidae. We focused especially on Cicadelline because they were more diverse. And we tested like 18 species in different studies, also one Cercopidae, and also some other species from groups that are non-xylem feeders that feed in other tissues like phloem were included in these assays to check the transmission. And we end up finding transmission only by the xylem feeders. So the xylem feeders were the only that transmitted, although we tested some species like Membracidae, Italionidae, Gyponini as vectors. And not all of the species were shown to be vectors in these particular transmission assays. Like some of the sharpshooters tested did not transmit because the transmission rate is probably very low, or because they don't like the host plant, citrus. Like Cercopidae is a grass feeder that we tested, so it doesn't like citrus. So we were not able to demonstrate the transmission experimentally. So this was experimentally demonstrated for 13 species of sharpshooters, most of them Cicadelline and 4 Proconiini. And these species are, most of them are very abundant in citrus orchards. So next step is to determine what of these species are relevant epidemiologically. So what factors determine vector relevance? So many things can be important. I listed some of these attributes that in the literature have been proposed, such as predominance and activity, natural infectivity, transmission efficiency, the host plants, if they visit host plants that are important for the epidemiology of the disease, especially those that serve as inoculum sources for pathogen spread. So starting with predominance and activity. Predominance is determined based on faunistic parameters like abundance, frequency, constancy in samplings, dominance. So predominant species are those that are very frequent, very abundant, sampled almost in any sampling date, and dominant species. Activity can be measured by movement of these insects in the field or visits, number of visits to the plants. So by yellows, this is just an example of a study done in northern São Paulo with yellow stick traps and trap plants. These were citrus plants sprayed with stick glue so that the insects would land on the citrus tree and would be stick to that plant. So you see in the same place, basically more or less the same composition of species were found, like Cometopia, Acrogonia, Gilobopterus, Bucephalogonia, which are very, very important species. And when you do these trap plants, more or less the same, although we found some grass feeder like Ferrariana also among the most important. Other attribute is vector competence, transmission efficiency. So you can measure how efficiently these insects transmit by estimating probabilities of transmission by single vectors So as you see in this table, transmission efficiency in citrus ranges from less than 1% to up to 30% in different studies. So it's generally low compared to what we see with Xylella fastidiosa, subspecies fastidiosa in grapes. In general, the first three were the most efficient in these assays: Macrogonalia, Bucephalogonia, and Gilobopterus. Okay, but also, besides the efficiency that you measure in transmission assays, it's important to check whether these insects, they naturally carry the bacteria in the field. And this is a result of their behavior of landing and feeding on inoculum sources of the disease. So you can do this by measuring the proportion of insects that are positive by PCR, ELISA, for example, or even better, you can do transmission assays. You can catch leafhoppers in the field, spittlebugs, and place them on indicator plants and see whether they transmit. We have done both, although I'm just presenting the results of ELISA tests showing that many of the species that were proven to be vectors experimentally, they also carry the bacteria in a sample, in a given sample. This is a nice graph published by Purcell and collaborators that show how transmission efficiency, natural infectivity, and population of the vectors, they interplay. So probability of infection can be a result of, is directly proportional to the number of visits, population, to the natural infectivity, and also to the transmission efficiency. So even with a relatively low transmission efficiency, like we find in citrus, 15% per insect, you can still have a very high rates of high probability of infection if you have like 10% of infective bugs in the field. If you have an abundant vector. With less abundant vectors, if they are efficient, that's kind of a trade-off. You can still have a high probability of infection. So this type of graph demonstrates these relationships. Host plants. So these vectors in citrus, for example, they have as a host the crops, affected crops, herbaceous weeds that are commonly found in the ground vegetation, and also wood hosts, trees and shrubs that are found in the natural vegetation. We have been studying this distribution of these sharpshooters from citrus in different plants. So some group you find very often on the citrus trees, like on Cometopia, Acrogonia, Gilobopterus, Homalodisca. There is some species like Bucephalogonia that are found both on the canopy and also in the ground vegetation, on the herbaceous plants, and some are very abundant in the ground vegetation only, not on the canopy of the citrus trees. So this group here, the first 5 lay eggs and develop on citrus. Whereas the grass feeders, they are rare on the citrus canopy, but they are very abundant on the weed vegetation. These weeds have been tested as possible inoculum sources. They have been sampled in the field to detect for detection of Xylella, and they have shown to be positive. They have been experimentally inoculated, and they can be colonized by the citrus strain of Xylella fastidiosa. But there are few efforts to try transmission from these plants to citrus. There was one study with Ferrariana, and this was not possible. There was no transmission. So the results, we don't have evidence that these plants serve based on the results with Ferrariana trevitata. Maybe we should do some other assays with other species. Concerning these trees and shrubs and wood plants in natural habitats surrounding citrus orchard. There are many natural habitats in São Paulo, like swamps, riparian woods, savannas, cerrados, and many have different plant species that serve as hosts of these abundant sharpshooters in citrus. So more than 40 host plants were found for the sharpshooters of different botanical families, and some of them were shown to be colonized by Xylella by mechanical inoculation. We have not done many samplings, but we never found them infected in the field. But we needed more studies to show this, more sampling. But these habitats, natural habitats, may have an important role in epidemiology or in disease ecology as a refuge and breeding sites for several sharpshooters. So when the orchards are sprayed, they go. You have a place for them to be protected and recolonize citrus orchards later on. And also they might serve as possible sources of inoculum and also genetic diversity of the pathogen. A model on how these different host plants and communities of vectors can work to generate this diversity is in this picture. So we have, for example, a vector species A that is very common in the riparian wood, and sometimes of the year it goes to the orchard when the plants are favorable for development and may carry a particular strain. You may have a species B that occurs in the other natural habitat and goes to the crops and may also carry another strain. And you may even have a highly polyphagous species that can move between the different natural habitats and among different crops and mix all the things. So different species can play a different role in these pathosystems. In grapes, we have more information about this type of studies in California, done by Sandy Purcell. So in this picture, I'm showing a situation, for example, in the north coast of California, where you have a major vector, the blue-green sharpshooter. You have this riparian environments with many hosts of Xylella. In that picture, we have Sandy Purcell. I visited a long time ago, and he showed me many plants that are hosts of Xylella. Sandy showed many aspects of that were very important for understanding of this pathosystem. In that particular situations, these vectors, they move from these riparian habitats early in the spring to the vineyards, and they carry this primary inoculum and establish these primary infections in grape, and you see a higher concentration of plants by the edge nearby these riparian woods. So this is a situation where the vector comes from every year from the woods. In another situation, we have the Central Valley alfalfa, which is a host of Xylella strains, and also vineyards. And you have a very clear movement of inoculum from alfalfa to the borders of the vineyard fields. In alfalfa, you have weeds, which are very good hosts of some vectors like green sharpshooters, red-headed sharpshooters, and they can acquire in alfalfa the pathogen and transmit to grapes. And primary spread is observed in this situation. I have done a sabbatical at Rodrigo's lab, and we did some experiments, and some transmission experiments explained a bit this type of primary spread. For example, if you use alfalfa as a source plant, the green sharpshooter effectively acquire and transmit it to grape. But it's not, transmission is not efficient if you have acquisition on grape or almond. So secondary spread is more difficult in that situation because green sharpshooter does not like almonds and grapes. not only the source plant can be important, but also the interaction between the source plant and the plant and the bacterium. For example, almond isolates did not multiply so well as the grape isolates. There's lower populations. And in fact, we get no transmission rate in this particular experiment. And very high transmission rate when you get the alfalfa as a source plant. But the isolates, because of their interaction with the host plants, they affected these results. Also, feeding sites can be important, not only the preference for the plant, but also where in the plant these insects will feed. So alfalfa has a higher concentration of bacteria in the base of the plant. So if you put the insects in small leaf cades or stain cades at the top of the plant and at the bottom of the plant, you get different results in terms of transmission to indicator plants. So the higher transmission rates, of course, when you place them in the base of the plant, because that's where the bacteria is more concentrated. And that has an ecological implication. Alfalfa is harvested very often, every 30 to 40 days. So for the bacteria to survive and be acquired, it must be in the lower parts of the plant. And the vector, to be effective, has to feed on that part of the plant. So vector feeding preferences and bacterial subspecies, strains, and interaction with host plants can affect vector competence. And bacterial strains should be able to colonize both source and the target plants. Another example, coming back to the citrus and to Brazil system, is coffee and citrus. Coffee and citrus are affected by citrus strains of, by Xylella fastidiosa, different strains. When you think about these two crops, they are sympatric, cultivated very often close to each other. They have common species of vectors that move between one crop and the other. So you would expect that Xylella fastidiosa from citrus would go to coffee and infect coffee and vice versa. But studies of molecular epidemiology have been done, like by Ovesio and his group, studying the population structure of this Xylella isolates from coffee and citrus in this nearby situation. And there was no mixture of citrus and coffee infecting isolates. Also, if you do reciprocal inoculations, putting citrus strains in coffee and coffee strains in citrus, you don't get cross-infection. Or you do get a bit. You can see like a local infection, but months later they disappear, so you don't have a chronic infection. So citrus is not serving as a source for coffee and vice versa, although they have similar vectors and are close to each other. So the body of epidemiological evidence have shown that citrus is a major source plant for spread of CBC. This is based on many studies, and we know today that both primary spread from nearby orchards and secondary spread from plant to plant play a major role in epidemiology. So because of this secondary spread takes place from citrus to citrus, it became obvious that those species that feed on the canopy, that lay eggs, that develop on citrus are key vectors. So this is an example of how these vectors can be determined. Now to go towards the end, just mention that we have this new problem in Brazil, in olives, a very similar disease occurring that is associated with Xylella. This association is still working on that, but it appears to be a problem there. And also in Argentina, similar reports. And the challenge now is to find do a similar work and find out what are the relevant vectors. So this starts again with surveying the community of xylem-feeders, with sampling by yellow stick traps, by sweep net, and determining prevalent species. For example, we have so far found 20 species of sharpshooters and one speedo bug, and also speedo bugs that are frequent in that situation, are predominant, so they might be involved in the epidemiology. There's one Crassoptera species that is that colonizes the tree. We find nymphs, so that may be important for secondary spread, for example. But we need much more information on, especially on host plant associations of these vectors, epidemiology. We need to search for hosts of both the vector and the pathogen. And also characterize the patterns of spread of the disease, both spatial and temporal patterns that can help. So to conclude this talk, I think in general, in any pathosystem, in emergent diseases caused by Xylella, you need a broad knowledge in order to determine this importance of vectors, of vector competence, prevalence, host plant associations, but also information from epidemiology, like how the disease progresses over time in the space. That can give very important clues on the source of inoculum. Vector colonization of affected crops is important for secondary spread, but not essential for spread among different hosts and habitats, as we've seen, for example, in grapes. And no colonizing but mobile vector species may still play a role in disease ecology. So we need to understand ecology of Xylella and vectors outside crops also in order to better manage these systems. Thank you.
Thank you, Joao, for the nice picture on the role of different vectors on different pathosystems. Now, the next speaker is Nicola Bodino from the University of Torino, Italy, with a presentation on phenology and population dynamics and host plants of Philaenus spumarius in Italian olive groves.
Okay. Hello to everyone. I'm pleased to present this study concerning phenology, population dynamics, and host plants of Philaenus spumarius in Italian olive groves in the framework of the EFSA project, collection of data and information on biology and control of vectors of Xylella fastidiosa. Okay, I will not spend many words on Xylella, but here you can see the current situation of Xylella in Europe with the subspecies Pauca in Apulia, the multiplex in France, and a mix of different subspecies in Balearic Island, just where we are now. The dots, the points, represent the olive groves subject of this study. So Xylella is a xylem-limited bacterium, and therefore spitterbugs that are the most common xylem sub-feeders in Europe are the natural candidate vectors. In particular, Philaenus spumarius is the only proven vector of Xylella in strain Codiro in Apulia. It is an insect species very widespread and highly, or even extremely, polyphagous, as it presents only one generation per year, and its pre-imaginal instar live protected in a foam spittle that they produce, while the adults are characterized by a long life and a summer ovaric diapause with ovipositions that occur only in autumn, at least in Mediterranean conditions. There are several other species of xylem subfeeders in Europe of the superfamily Cercopoidea or other superfamilies, but they are all theoretically possible vectors of Xylella fastidiosa, but are usually far less ubiquitous and abundant than Philaenus spumarius. Therefore, the aim of this study was focused on Philaenus spumarius and its, on some aspects of its biology and ecology, in particular population dynamics, phenology, nymphs and adult abundance, host-plant association, and sex ratio, and all this information will be used to create predictive models useful to design effective control measures to both slow the disease progression and assessing risk in non-infected areas. So the study was carried out on 4 olive groves per 2 years in different Mediterranean regions of Italy, in the north and in the south. So in the south, the Apulia region is a hot summer climatic condition, Mediterranean, whereas in Liguria is a warm summer climatic condition, so it's a little bit different. Furthermore, moreover, 2 olive orchards were located in the coastal areas and 2 in inland areas, so also different mesoclimatic conditions. The sampling methodology for nymphs consisted in 30 samples per olive groves in a quadrant of 0.25 square meter with a simple random sampling. And inside the quadrant, the nymphs were counted in a conservative way, so they were not killed but just lived in the field to not disturb the population dynamics over the season. All the host plants were identified and as well the plant community described inside the quadrant. The samplings were carried out on a weekly basis or 10 days. The adults were collected with a sweeping net and were counted always in a conservative way every 2 weeks. On the herbaceous cover, we carried out 30 samples of 4 sweeps each, whereas on the olive trees, 20 samples consisted of 10 sweeps each on the canopy of the olive trees, and on shrubs or wild woody plants, 10 or 30 samples of 10 sweeps each. Other xylem-feeders found during these samples were collected for subsequent identification, so non-conservative sampling. Okay, about the preliminary results. In Liguria, we found the nymphs hatching in early March, and disappear by late May, with a population peaks in mid-April, and the coexistence of different instars, the 2nd, 3rd, and 4th, during April, 2-3 weeks. We didn't find differences in Philaenus spumarius abundance between the 2 olive groves in 2016, but we found differences in 2017. This is probably caused by a different management during this year. In Apulia, the nymphs start to hatch in late February, early March, and disappear by mid-May, with population peaks in late March, early April. So the population dynamics was similar, but just shift a little bit earlier in Apulia, as we expected. It's warmer. Unfortunately, during 2016, the samples started a little bit later, so we didn't have a complete description of the phenology during this year. Okay, about the host plant preference, the nymphs were found on more than 140 plant species or genera, with Medicago, Crepis, Sonchus, and Trifolium being the most infested plants. But we found really important and changes in plant preference among different olive groves. This is due probably to differences in plant communities. So Philaenus tend to go on plants that he can find, obviously, and if it doesn't find a plant, it really easily switch to another plant species. So it's difficult to generalize. So it's also useful to analyzed the data at a higher level, at the plant family level, with Asteraceae being the preferred plant family, followed by Fabaceae. In total, more than 20 plant families were recorded as host, and it's interesting that the Asteraceae were really preferred with a 40% by early instar, the first and in particular the second. This is probably because the typical basal rosettes of Asteraceae that protect the early instar from desiccation. Okay, about the Philaenus spumarius adults, the population appeared to peak in Liguria in late spring and then again in autumn on herbaceous plants. Whereas on olive trees and other woody plants, the population appeared to be higher during the summer, or at least more constant during the season, the summer season. In Apulia, the population peaked on herbaceous host in late spring, and not always we observed a peak again in autumn. And whereas in olive trees and wild woody plants, there were higher population only during late spring and really early summer, with very, very low population during the mid-late summer on all the component, all the vegetational component of olive grove. About the preferred woody plants of Philaenus spumarius adult, the wild woody plants, we observed a clear preference for oaks and lentisk in Liguria and for myrtus and lentisk in Apulia. But this is really dependent on the plant communities found in this different region. That is clearly quite different. So, summarizing these preliminary results shown before, we quite agree with Weaver and King that already more than 60 years ago were saying that they are able to survive on almost any plants that provide them sufficient moisture to maintain their feeding habits. Indeed, we found no clear preference for specific plant species, but instead an important influence of plant structure, with especially, as I said before, basal rosettes that probably give protection with an optimal micro-niche environment for temperature and especially for humidity for this delicate early instar. But we record also a quite clear host plant shifting during the nymphal development, different families, but also different species that, and we will analyze later. And this host plant shifting is probably influenced by instar of nymphs and maybe different needs, but in particular phenology and availability of water supply of the host plants. They change as well as soon as the host plants become no more favorable. The stage structure population dynamics showed a co-occurrence of several instars, second, third, fourth, for 2-3 weeks, and this is probably caused by a delayed egg hatching influenced by micro-niche conditions. Temperature and humidity can drastically change also in a space of few centimeters. Population densities of Philaenus spumarius appear to be influenced by several factors. Among them, the climatic condition at the macro and meso level, plant communities, and agronomical management of olive groves and surrounding areas. For the adults, there was a clear seasonal movement between different vegetation components, determined mainly by water stress of host plants and also the oviposition needs that bring at least females, but also males, in the ground vegetation in autumn. The difference in population density between Liguria and Apulia that we observed can be at least partly explained by the water stress that was in general minor in Liguria, and therefore we found a higher and more constant population of vectors on olive trees during summer, whereas in Apulia, with a complete drying of herbaceous host and also a more severe water stress of woody plants, caused a low lower population of vector on both herbaceous and olives. And this situation can trigger migration on a larger scale of Philaenus that go looking for more suitable host outside the olive grove. Concluding, obviously, Philaenus spumarius seasonal migration and variation in population abundance across the Mediterranean region can have a relevant influence on the pattern and speed of the spread of Xylella. So this study provides useful information on vector phenology, population dynamics, and host shifting that can be used to develop predictive models of the vector population density, useful to design effective control measures. And we need also further study on a finer scale on vector biology and landscape ecology that will allow to improve these models. If you're interested, there are two posters, one on host preference by Crescenza Don Giovanni and one on dispersal capabilities of Philaenus by Elisa Plazio and her colleagues. If you want to look for them and ask, feel free. And I want to thank you for attention, and thank you also EFSA for its help and funding the project. Thanks.
Thank you, Nicola. You can sit here. The next speaker is Daniele Cornara from CSIC, Instituto de Ciencias Agrarias in Madrid. That will present an EPG study on Philaenus spumarius feeding behavior.
Hello, everybody. And I'm here to present to you our work about Philaenus spumarius feeding behavior and transmission mechanism of Xylella fastidiosa. So basically, let's start this talk with a question. Why is it so important to study vector feeding behavior? So, in particular, the feeding behavior of our vector of Xylella fastidiosa, Philaenus spumarius. And let's try to reply to this question just looking at what we know about Xylella fastidiosa transmission. So, the transmission of this bacterium has not a latent period. Xylella, once acquired by the adults, is persistently retained in adults. Xylella is neither transovarially or transstadially transmitted. Once acquired, the bacterium is retained, bind, and multiplies into vector forget. And there seems to be no vector strain specificity. Eventually, as remarked by Professor Joao Lopes, a key element for the transmission of Xylella fastidiosa is vector behavior. And basically, when we talk about behavior, we are talking about two stuff. The relationship of the vector with the host plant, so basically host range and preference for certain tissues and so on, and we are talking about vector feeding behavior. Characterizing vector feeding behavior will furnish us elements to understand the transmission mechanism of Xylella fastidiosa that has eluded researchers for more than 70 years. But, so, another question, how to study the feeding behavior of an insect that has a probing mouthparts, so basically the stylets are invisible from outside while the insect is probing? Well, the most powerful tool we have is EPG. EPG means electrical penetration graph, and basically with EPG, we make the insects and the plant part of an electrical circuit. Then we use biopotentials and electrical resistances that transform the constantly applied electrical input in a variable voltage output that is graphed in waveforms. Basically, to be the more clear as possible, each waveform is a phase of the probing, okay? It's a step of the probing. So what have we done in Madrid? We characterized the feeding behavior of Philaenus spumarius on olive, cultivar picual, and veg plant, both of females and males. And then we looked for the biological activity, so the biological meaning of each one of this waveform by CT scan microtomography, so looking at the position of the stylets during each one of the observed waveform, basically reconstructing the 3D image without excessive manipulation of both the insect and the plant, and by video recording DPG, looking at in real time the excretion of the adults and the excretion and secretion of the nymphs with the formation of the spittle. Okay, these are the main waveforms. We detected 5 main waveforms for Philaenus spumarius that we named C, XC, XI, both high frequency and low frequency, and R. Just now, look at the likelihood of the events. NP means non-probing. When the insects start a probe, we usually observe waveform C. Waveform C for Philaenus takes no longer than 10 minutes, usually 5-6 minutes more or less. Then the insects pass to XC. Takes 2 to 6 minutes and pass to XC. For Philaenus spumarius, Philaenus spumarius spend more or less the 70% of its probing time in XC, and then repeatedly alternate during the probe XC and R, then from XC or from R parts to C and interrupt the probe. And usually for Philaenus spumarius, in an 8-hour recording, we observe an average of 3-4 probes. This is the likelihood of the events. Now look at the biological meaning of each one of these electrical signals, each one of these waveforms. These are the result of the CT scan microtomography. So what have we done? Basically, with the CT scan, once we observed the waveform of interest, we interrupted the feeding by liquid nitrogen, then we analyzed the plant tissue and the insect by CT scan microtomography, and we reconstructed the 3D image of the stylet inside the tissue without manipulation of it. And these are the results. The stylets are in xylem vessel during waveform XC, XI, and ON. During waveform C, both the stylets and the salivary sheath are outside of the xylem. During waveform ON, the salivary sheath is in xylem, while we observe the stylets partially withdrawn. Let's look now at the excretory behavior and for the nymphs, excretion and secretion. What have we observed? Excretion, and for the nymphs, secretion, excretion occur only during this waveform, the waveform XI, that is characterized by this peak and this plateau. And by analyzing the droplets excreted with reaction with 93.2%, we basically observed that Philaenus spumarius feed only from xylem, in just xylem sap. But interestingly, if the video starts, that's always tricky. See? It's always tricky. No, trust me, I have the video. Okay. Okay, I'll try. So, by looking at the nymphs, and trust me, we have a video that we are going to publish soon. So, through the tenuous cuticle of the nymphs, you can see the pumping, the movement of the cybarial pump of the nymphs, and we correlated the movement of the cybarial pump with the shape of the waveform XI. So, each one of these peaks corresponds to a contraction of the muscle, while each one of these plateaus corresponds to a release of the muscle with sap swallowing. So, these are our conclusion. This is the biological meaning of Philaenus spumarius. This is the feeding behavior. Waveform C means stylet penetration building of a salivary sheath while the stylets are moving toward xylem. And in this, Philaenus spumarius is extremely quick compared to other xylem feeders, reach really rapidly the xylem. XC is the first contact with the xylem vessel with a trial ingestion, and generally takes from 2 to 6 minutes, and then the insects pass to xylem ingestion. So waveform XI, both high frequency or low frequency. But loading the cybarial pump to extract xylem sap requires a lot of energy. So the insects need to rest and alternate period of sap extraction with period of resting that are waveform R. Waveform R is basically flat waveforms, that means resting. Then eventually waveform N. So our explanation of waveform N is that is an interruption during XC or XI, basically it's xylem phase, but we need more research efforts exactly to understand what a waveform N means biologically. Now, let me show you a couple of ongoing work we are doing in Madrid. First of all, artificial diet. The problem is this: a lot of authors reported that Philaenus spumarius refuse any kind of artificial diet, and that's a problem, because of setup of an artificial diet system would be really useful for both feeding behavioral studies and transmission of Xylella fastidiosa, as shown in Sharpshooter by Rodrigo Almeida and Chiellini in 2009. So basically in Madrid, since more or less 6 months, we are trying to set up an artificial diet combining different kind of diet with different concentration of amino acids. And we are also trying to set up a tool to deliver the artificial diet to Philaenus spumarius. We have a couple of promising candidates, and we will keep you updated as soon as possible with this, as we will keep you updated with our preliminary results about transmission mechanism of Xylella fastidiosa. Basically, after the characterization of the feeding behavior of Philaenus spumarius, we want to know which one of these waveforms, so which behavior, is correlated with the transmission of Xylella fastidiosa. And after 70 years, we don't know which behavior is correlated with the transmission of Xylella fastidiosa. So this is part of a preliminary study we carried out in Racale in Italy during August and September 2017, thanks to the collaboration of CNR and of Bari. Basically, we collected Philaenus spumarius, infected Philaenus spumarius. We caged them for an acquisition access period on olive of 7 days. Then we connected to the EPG, this is an EPG-assisted transmission studies, singly Philaenus spumarius, leaving them on olive seedlings for a brief inoculation access period. Basically, we interrupted the probe once the waveform of interest was observed. And basically, we tested waveform C, waveform XC, waveform XI, and waveform N, both occurring during XC and XI. Basically, we want to know which one of these waveforms lead to the transmission of Xylella fastidiosa. Then we moved the insect, Philaenus spumarius, to a periwinkle plant for an inoculation access period of 1 day, and we left the plant for 1 month for an incubation period. Then we will test both the insect and the plant by qPCR for the presence of Xylella, and regarding the plant, both the inoculated tissue and the part distal to it, to see if there is or not systemic infection. And this is preliminary work. We will continue it during 2018. Eventually, we as Madrid Group, Ferrer's Lab, would like to thank people that helped us a lot during transmission tests in Italy. So CNR, Bari Group. We would like to thank also Cooperative Acli Racale with Enzo and Federico Manni. We would like to acknowledge the organizer of this meeting, and we and I basically would like to thank you for your attention.
Thank you, Daniele. Next speaker is Miguel Miranda, our kind organizer from this university, and his presentation is on seasonal pattern hosts and abundance of the potential vectors of Xylella fastidiosa in Mallorca.
Thank you, Alba. Thank you, Domenico, for this kind introduction, and thank you as well to the scientific committee for giving us the opportunity to present these preliminary results on the vectors in Mallorca. As you see, there is a lot of people on this presentation, and I want really to point out the work of Alicia Marquez, Tony Sureda, and Armando Amore, that contributed very much to the field work of this study. So, well, Blanca Alanda presented brilliantly yesterday the situation in Mallorca. This is just a reminder that we are dealing with, I think, a particularly unique situation where we have a lot of different subspecies and in the different islands. In Mallorca, you see fastidiosa and multiplex, pauca in Ibiza, and multiplex as well in Menorca. So since the detection of the disease in 2016, we started a really preliminary project to try to identify not the vectors, but at least the fauna that could be related to the transmission of Xylella. So the project started in March 2017, and basically the objectives of this project were very, as you see, the basis, as Joao was pointed out, just to better know the different families of insects that could be transmitter of the bacteria. And because of the time we had and the resources we have, we focus only on what we knew already from Italy. So we focus only on the Afrophoridae. These are the results that I'm presenting here now. So we study the species and also the seasonal pattern. And finally, I will present some of the preliminary results of the bacterium in the vector by using molecular tools. So really, all these groups of insects are really poorly studied in the Balearic Islands because they are not a pest. They are very common insects that's been always around, but never causing a problem that deserved any kind of control. So nobody studied as well, neither the phenology or neither all the species that are present in the Balearic. So if we refer to the previous report from EFSA 2015, 3 species were already recorded in the Balearics, which are Philaenus spumarius, Neophilaenus campestris, and Neophilaenus lineatus. So even before finding some of the problems in different cultivars, like for example in vineyards, we started already in March to include all the very important cultivars in the Balearics in our study. So we decided to select 2 orchards or crops from citrus, almond, olive, and vineyards in different parts of the Balearic. The most important thing was that the farmers do not apply insecticides, and also they do not manage the cover of the plant. I mean, we wanted really to have the plant coverage there, the herbaceous cover, to really have to follow the cycle of the insects. And we also, in collaboration with the Conselleria, with the local government, we also selected 3 sites that were very nearby the positive sites where the disease was detected in October 2016. So this was our sampling sites. Here you have an example, like this one is vinegar, citrus, almond, and olives, all with plant cover except the vinegar that it was impossible, almost impossible to find vinegar where they didn't manage the cover. So you will see that we have results of the plant cover, but not from vinegar. For the capture of insects, we used different methods, as Nicola was also pointed out, and this work was conducted in collaboration with Dr. Alberto Ferreres. We designed a protocol to sample the insects in the different crops, not just in olives, but also in vinegars and citrus and almond. And for this, we decided, of course, to to use direct observations of the fawns in the plants. We also use the sweeping net, that I think is the most useful tool to capture this kind of insects. We also use bags, trying to use bagging on the branches of the different trees. And we also used backpack vacuum to aspirate the insects. Here is a summary of the sampling methods. Of course, not all the sampling methods are okay for all the crops. So you will see here that for herbaceous, we use the fawn sampling and sweeping net. For olive trees, we were using the sweeping net and the bagging. Here you have the scheme and the number of samples we were collecting from each of the crops. And for almond, citrus, and vinegar, because of the structure of the branches and of the vegetation, I mean, the sweeping net is not possible to be used there, so we were using just the vacuum. So these preliminary results we obtained, I would say that it coincides very much with the results you saw from other places, of course, in the Mediterranean region, like Italy, for example. So we found the foams basically in April. You could find some of them in early March, but most of them were, in this particular year, 2017, were concentrated in the month of April. And as you see here, olive crops were the ones that contained more number of foams. But compared to other studies in Italy, really our abundance of foams, and also you will see adults, are very, very low. In regards to the plant host families, as you see here, 90% of the families that contain the foams are from the family Asteraceae, and here you have the species, different species, very common in all our orchards, abandoned orchards, or even managed orchards. And this is very important, as Nicola was also pointed out, to know really the landscape distribution and all the plant communities to really understand how Philaenus or other species are feeding on this kind of environment. Here you have also all the plant species and the percentage of foams per plant host. And you see that some of the species are very much more, they got more foams than others, and particularly species like Riccardia pricoides. Most of the species that we found, they were hemicryptophyta. That means that they just the aerial part of the part die during the summertime and autumn time, depends on the cycle of the plant. They may have different floration times, or flowering times. And this is for sure a resource, a type of plant that this kind of insects like very much, because they keep some of the resources for the next season. So if you see here now, we separate the same species but by crops, and as I was mentioning before, the olive crop was the one that hold a higher number of foams, and particularly this species again, Riccardia pricoides, is again the species the plant species that we detected the higher number of foams. In regards to the positive place, the place where Xylella fastidiosa was detected at the beginning, then as you know, it was detected everywhere, but at the beginning it was just in these areas, and we focus in these areas. So you see also that the foams were also present there, but not in this place in San Llorenç, because again, very similarly to the results that were presented today from Italy, there is some difference in temperature and the foams at this place, at this particular time when we sampled in April, were already completed already the cycle. In regards to the plant species in these positive places, as you see, we have the same species as in the other places. Riccardia pricoides again is one of the species that had the higher number of foams, but differently from the other places, non-bolifera, Phoeniculum vulgare species that is in our area is always holding a lot of foams and is really particularly used by Philaenus spumarius for its development. In regards to the adults, I would say that our results are quite sad because we were expecting that we will use the sweep net and kilos of Philaenus spumarius we will capture in our sweeping net. And we knew from other places like Italy that Philaenus spumarius is really highly abundant, for example, in olive trees. Now we see that it could be also depending on temperature. But really, I don't know if it's this particular year because, you know, the results are only from March to now, to November. But really, the density of adults in all the crops was really, really low. It means that from an epidemiological point of view, we have no clear indication that we have a really abundant vector that could transmit, or maybe is very efficient and it's been a long time transmitting the bacteria. Okay, in regards to the cycle, we found basically May with a peak of abundance as you see in olives, but very low adults, and also now in October, November, but no adults in July and August in any of the orchards. In regards to the species, we confirmed the 3 species that were already published in the EFSA report, Philaenus, Neophilaenus campestris, and Philaenus spumarius. In regards to the abundance of adults in the herbaceous vegetation, as you see, the numbers are low, except in some cases that we capture 20 adults of Neophilaenus campestris, but in spring and in October, the density is again lower than in spring. In regards to the species, we just have Neophilaenus lineatus as a new species. And here is more or less the life cycle. It's very similar to what we see in other Mediterranean areas. And really the key issue is now this. What are they doing in summertime? And they are not in the crops. There are no plants there are no cover plants because they dry. In all the Balearic Islands, they dry out. So we really need to find out where do they spend the summer, if they are in a kind of estivation or what. And in regards to the molecular detection that is conducted by the laboratory here in the Balearic government, there's not much to say because we got just around 39 specimens, and they were all negative to PCR. So we are still really don't know. We have no any indication that what's really the vector here in the Balearics. So in conclusion, you see what I was saying, that the highest number of nymphs was found during April, in spring, as you saw. There are several species that really had the high number of nymphs. In summer, we don't know where the adults go. They are not in the main crops, for sure. And in autumn, the adults were detected again in the main crops. And they were more abundant in the plant cover, but still in low populations. And in general, Neophilaenus campestris, it seems to be much more abundant than the other species of Afrophoridae. So I just want to finish acknowledging to the owners of the orchards that allow us to conduct these studies, of course to the government of the Balearic Islands for funding this preliminary project, and also to all the people that contributed in one way or another to this work. Thank you very much.
Thank you, Miguel. Next and last speaker of this first part of the session is Anna Markheiser from the Julius Kühn Institute in Germany, with a presentation on significance of xylem feeding Auchenorrhyncha in orchards and vineyards in Germany.
Thank you, Domenico. First of all, I want to thank the organizers for a very warm welcome here in Mallorca. We had 3 degrees this week, so I'm Happy to be here. As you may all remember, Xylella fastidiosa was detected for the first time in Germany in 2016. Potted oleander plant was infected with the subspecies fastidiosa. Buffer zone was arranged and no positive vector or plant could be found under open land conditions so far. This case leads us to the question, how could Xylella be spread and established in Germany? Our scenario for risk assessment is that a positive plant is introduced into a private garden or park. This could be ornamental plants, which are often imported from the Mediterranean region. If the bacterium is ingested by a xylem-feeding species, and this species is polyphagous, it could be transferred to ejected woody host plants, which show a more tolerance to climatic conditions. And this could also be an orchard or a vineyard. This is why we ask ourselves, how is the potential vector situation and host plant situation of these xylem feeder species, independence of the habitat type. We started our surveys in April this year, and we did a monitoring of spittlebug's and other xylem feeders in 4 habitat types. We observed 22 locations in 2 wine-growing regions in Germany, which are called Mosel and Palatinate. It's located in the southwest. And one example of such a location is our institute, where 3 habitat types are arranged nearby. In the middle you can see a garden, 200 meters apart, wild vegetation with woody plants, and direct beside a small vineyard. This reflects an authentic situation for a dispersal of potential vectors. To give you an idea of the chosen habitat types, here you can see a sherry orchard, a vineyard in the wine-growing region Mosel, a wild vegetation, as well as a private garden. We did our sampling according to the conventional methods of our X of Actors project partners. We did a visual observation of nymphal stages of spittlebugs on the ground cover to identify the host plants. I will not go into detail. I think you heard about it a little bit, and if you are interested, you're welcome to visit us at the poster session. We installed yellow sticky traps for primary assessment of present xylem feeder species. And in addition, we collect adults with sweeping net on the ground cover, as mentioned before, and as on the canopy on selected shrubs and trees. We did, as mentioned before, a defined sampling, so we compare our results to other project partners. According to literature, Germany has 26 species of xylem feeders out of 4 families, 13 species of Aphrodina, like Philaenus spumarius, Aphrophora alni, 6 species of Neophilenus, as well as Leperonia coleoptrata, for example, 6 species of Cicadellina, like Evacanthus interruptus, Cicadella viridis, and Raphocephala fenae, 4 species of Cercobidae, and 3 species of cicadas. If we come back to our example of location Geil-Wallerhof, we can see the distribution of the xylem feeders within an habitat type. Visualized is the mean number of catched adults per yellow sticky trap. In this home garden, we could only find the species Raphocephala fenae, which was present with July. And here you can see only 3 species, for example, and we not do show all. On the wild vegetation nearby, we found all these 3 species, whereas in the vineyard we only found Philaenus. Interestingly, Craphocephala fenai, its nymphal stages are monophagous on rhododendron. This is a plant species which was not available in the wild vegetation. This implies that the adults must have been emigrated over a distance of 200 meters from the home garden to this wild vegetation. The next slides give you a summary of the present xylem-feeder species independent of the habitat type, as well as they were on the ground cover or on the canopy. In the home gardens, on the ground cover, most of all we find Philaenus, spumarius, and whereas in the canopy the most present species was Craphocephala fenai. If we compare it with wild vegetation, which could be situated beside, we found also the most prevalent species on the ground cover Philaenus, whereas in the canopy dominant were the species Aphophora alni as well as Philaenus spumarius. Interestingly, we couldn't catch any individuals of Craphocephala fenai, although we catch some individuals with the yellow sticky traps. This depends on the density of the individuals in this habitat type. We see other xylem-feeder spectrum in orchards. There were also available a large number of Cicadella viridis as well as Aevacanthus interruptus on the ground cover, but still in the canopy present were the both species Aphophora alni and Philaenus spumarius. If we compare it to the vineyards, for example, we can additionally find on the ground cover Cecropus species, most of them on Medicago, and on the vine canopy we can find most of all 3 species of Philaenus beside Aphophora alni and Neophilaenus species, most of them Neophilaenus campestris. If we compare the xylem-feeder spectrum of these 2 wine-growing regions, Mosel and Palatinate, in the vine canopy, we can see that most of all Philaenus spumarius was present in the canopy, whereas in the wine-growing region it looks a little bit different. We had beside Philaenus, Aphophora alni and Neophilaenus species. And this example shows us that we have regional differences in the xylem-feeder spectrum as well as in the density. To give you an idea of the dispersal of xylem-feeders within one habitat type, I will show you an example of a vineyard in the wine-growing region Mosel, where we observed high population levels of Philaenus, high population levels for Germany. We found the first adults at the end of May, and they began to spread from the ground cover to the vine canopy with the mid of June. And June is the month where we observed the highest population of Philaenus within the vineyard. And afterwards, they began to spread and move back to uncultivated ground cover, which is located beside the vineyard with the middle of August. And Philaenus was present until the beginning of October. To give you an idea of the seasonal development of the xylem-feeder species in our region, the spittlebox developed by the end of May, the nymphal stages, and 4 weeks later we can see the adults, and they were present in the habitat types until the beginning of October. Cecropus species were species which we found the earliest with the beginning of our studies, and leafhoppers like Aevacanthus interruptus and Aevacanthus acuminatus developed earlier than the spittlebox, but had a shorter life range if we compare it to species like Xylella viridis and Graphocephala fenayi. Here you can see the occurrence of these species in these habitat types. And important for you is that we had 4 species which were present in the home garden, which means they could be an interface from the introductory host to the establishment host. And these are species like Philaenus spumarius, Aphorphora alni, as well as Xylella viridis and Graphocephala fenayi. We also identified some host plants of the herbaceous cover and species like plant species like rosemary and lavender, which could be infected with Xylella irrespective of the species. We found only Philaenus, and we also identified some woody host plants. And the important thing I want to say here is that Prunus avium as well as Vitis vinifera are plant species which have a high spectrum of xylem-feeders, and this means that they are a high risk for an import of the species to our country. Well, Xylella fastidiosa, is it a risk for orchards and vineyards in Germany? Up to date, establishment in Germany is unpredictable. We don't know about the tolerance of the bacterium to our climatic conditions. We think overwinter survival under protected cultivation is conceivable, and massive woody host plants could be responsible for a permanent establishment. We found potential vector species which may connect the introductory host with orchards and vineyards, like the example Graphocephala fenayi, and a further dissemination of the bacterium could be followed by secondary vectors, and that could be species which are polyphagous like Aphorphora alni and Philaenus spumarius. Our topics for the next season is collecting information on the vector occurrence in other straits of Germany. We want to know more about the significance of phloem-feeders as potential vectors. We found also some phloem-feeders in the tree canopy, like cherry, which could also maybe transmit or occasionally xylem-feeders and transmit the bacterium. We are actually analyzing the feeding behavior of Philaenus on different host plants together with our partners in Spain with the EPG system. Yeah, and we do further evaluation of host plants in Germany. So thank you to the colleagues, and thank you for your attention.
Thank you, Anna. So I want to thank all the speakers, first of all, for their nice presentation, and also for keeping their presentation on time. So now we have about half an hour for discussion. So discussion is open.
Thank you. For all the speakers. So now we are collecting data on the spittle bug in Europe. This project funded by EFSA was focused on data which can be used in risk assessment, other in understanding the biology, but there's a lot of data being collected. What is the next steps in order to have IPM strategies for spittle bugs, which before were never considered in our defense plans?
Who is willing to?
Yes. But I would say that probably now the management that is already done in Apulia region is quite effective because it goes to affect the nymphal population. that it's clearly the stage more easily controlled for obvious reasons. They are not able to move as easily as adults, so try to lower the nymphal population by cutting the grass, mowing. I don't remember the term. Tillage. Thanks. Probably, I guess that is one of the best options to keep the population of the vectors, of the spitterbugs, low.
If I can add, a possibility is also to look at the plant species composition of the grass cover in the different crops, and maybe trying to favor those plant associations that are less suitable for the vector. I think that in such a way we can naturally suppress the vector population to more acceptable level. But this is highly dependent on the crop, on the specific situation. I don't know if you have that.
Can I? Yeah, but I guess, Giuseppe, you were asking more, what do we need to know to really set up a risk assessment, for example, or integrated pest management? Well, I think that probably one of the most important steps is to really understand what is the role of each of the species we are dealing with. I mean, there is no point to try to recommend generalized IPM methods if we don't know really the role of the different species and the species composition we have in every place. Because in some cases, some of the approaches could be in one way, in some of the cases could be in another way, just because, as we were saying here, and we saw the species composition of plants is different, and also that could also influence on the species composition of the vectors. And then you may have vectors which are very low competent on Xylella, and depending on the situation, you could try to do other kind of IPM. So to me, and this is one of the things we are for sure going to explore the next years, is to try to understand what is the role of each of the species on the transmission. Not just to put all of them in the same bag, because then IPM makes no sense.
It's active? Okay, so maybe I think like this because I work in a group that is more interested in vector-plant pathogen interaction, but as remarked by Rodrigo Almeida, Joao Lopes, each area, each specific area has its own vector, each crop has its own vector, so transmission tests are essential, like Philaenus spumarius is the main vector to olive in Italy. Philaenus seems not so important in transmission of Xylella st53 to olive in Italy. But here on another crop, I don't know. In France, I don't know. So looking at more the interaction to detect for each crop in each area, what's the vector, it's essential. That's essential.
So, okay, we can...
I wondered if anybody has any data from nighttime activity. Do they change host spent at night or this kind of thing? Some leafhoppers do. They're in the grass all day, and like most good entomologists, they go home when it gets dark. But this insect flies from the grass in the trees. All the adults do, and then they're back at sunset. So our California species on grasses fly mostly for about 30 to 40 minutes right at dark. So it'd be useful to know that. And what prompts this question is they're not attractive to yellow. I don't think anyone's brought that up. And there's a lot of you using sticky traps. Do you know what colors are most efficient and how that would compare to landing on vegetation, for example? So any comments?
Yeah. We used most of all yellow sticky traps, but we observed that we had also traps which were more colors included, and we found blue and so on, blue and red, I think, and we found Cercospis species which couldn't be found with yellow sticky traps, and I think this could be an aspect. And in addition, in another project where we are involved with Flavosaurus doré, we are observing other vectors which are not collectible with a sweeping net, but we found it on the yellow sticky traps. Maybe they are night active. That could be possible.
I think that one of the slides that you show, Anne, about Graphocephala fenai, you caught quite a large numbers of Graphocephala in the yellow sticky traps, but in the same area with the same sticky traps, you didn't catch any kind of spittlebugs. Is that right?
Yes, in this area we couldn't catch the Philaenus with spittle. It was more effective with the sweeping net than the yellow sticky traps.
Well, that suggests actually what you were talking about. If they are night flyers and they are not attracted by the yellow color, but the Graphocephala is, so it could be nice to test that hypothesis.
Yeah.
I was going to add that sharpshooters in general are highly attracted by the yellow traps, but the Cercospis or spittlebugs may not be. So this type of sampling methods are very important to use different methods and to have an idea of these different species and activity. And maybe if the night flight, sometimes the light traps can help too for this.
I mean, we have a lot of experience on light traps, and I never found any Cercospis inside light trap for Lepidoptera or mosquitoes, Culicoides, UV light traps, white light traps, any kind of light trap. And we never found maybe some Cicadelidae, some or maybe some even some other Homoptera, but I can't remember in all my years of light trapping any Afrophoridae or Cercospidae. So, well, but this is a matter of interest. Maybe they are not purely night flyers like all these insects that we were mentioning now, but they are crepuscular flyers, or they change the activity during the sunset, and this is for sure worthy to explore.
Hi, good morning. Congratulations, everyone, for your work and for your presentations. Thank you very much. It seems clear that in the Balearics here, there's quite a unique and complex situation with an infestation with multiple strains of Xylella. And at the same time, Dr. Miranda was saying how there's been a very low amount of spumarius insects found, and that those that have been tested have given negative to Xylella. And also the colleague from Madrid was saying how in 70 years there's still no assurance. They don't know yet how Xylella has passed from insect to plant. So my question is, can it really be assured that the vectors being studied are the only or even the main cause of the problems in the trees? Is there any investigation being done into other possible causes? Because if we're looking for answers to a global problem, perhaps we need to take a step back and see if that is the single root cause of the problem, or are there other causes that would need to be studied as well? I wonder what you all think about that.
You mean other ways of transmission?
Yes.
No, I mean, any of you, I think you could answer that, but I mean, a part of the insects and when you use grafting, there is no other major way of transmission. I mean, the main way of transmission are insects for sure. I think probably the only.
Yeah.
However, it seems that the data doesn't really point in that direction. If the trees are dying, and Xylella is one of the living beings that are present there in those trees that are dying. Firstly, is it the only cause of the death of the trees? And secondly, are the insect vectors the only reason why that bacteria is transmitted? Can we really, really say that?
Yes. Yeah, the
Based
answer is
on-
yes. It's just
I would really like to have more data about that we can provide people who are asking those questions. I work with farmers in the field, and they don't see that as the reason why their trees are dying. They're being told that is the reason, but
I mean,
many of them are questioning that.
We are discussing now about the transmission. I mean, about the disease itself, and we are now talking about the vectors and disease. We are not talking about now trying to discuss if it's Xylella, what is killing the trees or not. I mean, I think this is clear for most of the people here. So then, if you want, we could discuss later on about that. But now I think it's not really the moment to discuss if it's Xylella or other things that are killing the trees, honestly.
I really congratulate you on the work, because it's essential work, what you're doing. I'm just wondering whether
it's
insects...
It's a really preliminary work. I mean, as you see, we just analyzed 40 insects. This is nothing to really understand how is the pathosystem of Xylella in the Balearics. We just started in March, so I really beg your patience and just to wait maybe one more year, and we will have a more clear idea of what's really going on in the system, in this particular system in the Balearics. Thank you.
Thank you.
Okay. Rodrigo or someone else? Okay.
I don't want to get into the merits of the disease. It's a different topic, as Miguel brought up, but these insects are the vectors, of Xylella. We've known that for almost 70 years, and we understand a lot about the transmission. What we don't understand are specific probing behaviors associated with inoculation. So we understand a lot about how it's transmitted. We understand who the vectors are. It is possible that 70 years worth of science are not completely correct. That's always, the case. But there's been an immense amount of work that has been done in the United States and Brazil independently, and who the vectors are and how it happens is really well understood. But there is always more to learn, and that's what Daniele was referring to.
Yeah, and another point about if insects are or not the vectors is that once you see a tree which is not covered by insects, doesn't mean that the insects are not transmitting the disease. So there are many examples in the literature, for example, for non-persistent viruses, where you don't see the aphids, but they just probe, land, and in few seconds they transmit the virus. I'm not saying that Xylella is the same, but could be in a quite similar way, that you don't see the vectors in the tree, but they can just transiently stay for minutes and go to another source, and they will leave the virus there. Okay, so if a farmer doesn't see the insects in the tree, doesn't mean that there is no transmission.
One way to answer this type of questions, you can cage plants, for example, with nets, and you see you notice that those caged plants will not be infected. So this type of experiments have been done, for example, in citrus, and when they cage plants, they don't get infected. So suggesting that you have an airborne vector. And populations are low, but even if very low populations, even if you don't sample a lot the plants, you'll find very low populations. Even so, the vectors can be important because they can be efficient. And you are talking about perennial crops. the number of visits these plants receive over years is quite significant. So you may have 1, 2, 5, 50, and not infected, but there will be one infected insect that will land eventually and will inoculate. And you may have several inoculations over several years.
Okay. Hi, I missed a little bit some critic voices about what's happening here. We are here in Mallorca because there has been detected the Xylella fastidiosa 1 year ago. But indeed, the problem is that we have Xylella fastidiosa since 2003. There's an outbreak of almond leaf spore disease from 2003, and was confused the disease was a fungal disease. And we have now about 1 million trees infected with Xylella, only on the almond trees. On the wild olives, we have probably 1,000 other trees, and the fig tree, we have a big problem. The thing is that if we have an almond leaf spore disease with 76% incidence, you must know that the infection, the transmission, should be from tree to tree. And it's very, sorry to say, it's a bit sad that 1 year after, we don't know which are the vectors. And this happens because there have been a lot of discussion here in Mallorca about which is the widest spread of the disease. And indeed, there have been people from the European Commission here, from the Spanish government, from the local government, that have been discussing about, through 1 year, about if we have to do some contingency measures or eradication. And I think I want to say that this is nonsense, because if they had listened to the right people, people who know something about plant pathology, this will not happen. We will be here talking about the vectors. We will be talking about some data from the genome and other things. Just I want to point this out. Sorry.
Miguel, do you want to comment on this, or?
No, I have nothing to say. Thank you. Giuseppe, he has.
Yeah, I'm sorry. This is a question, a different question, not about transmission. But I'm not an entomologist, but I always ask about, is the insect vector suffering a kind of ill or disease by having this Xylella in its body?
Xylella interacts with in the foregut of the vectors and develops like a biofilm in the pre-Siberium. The channel is kind of narrow, so sometimes may affect a bit the movement of fluids, maybe. But because it's not a circulative pathogen, not multiplies in the vectors, usually we don't expect like a pathogenic effect of Xylella, except for maybe some difficulties for ingestion. But the infective bugs, they live for a long time. There are transmission experiments that have been done for assessing persistence, so they can, bugs infected, they can transmit from 1 day after to up to 2 months later. There are data like this showing that no pathogenic effects.
A question, what is the way to study if and how these insects are moved by the wind? So it's if they are moved by the wind, if they fly with the wind, which distance they can, this insect can spread, because that's important to understand how much you can spread an epidemic from a starting focus at the start, of course, of the epidemics. Is there a way to study this?
Yeah, by the wind. I mean, you have all the data from, for example, several aerial traps, the Roth hamster traps, that I'm not familiar with but there is people here, for example, from Sasa, from Scotland, that they have there one of those traps. And these traps are very much used for aphids, for example, and they just record all the aphids that move by wind. I don't know if this could be, maybe Alberto, you know more. I don't know if this could be also used to sample these kind of vectors, which are much bigger than aphids and all these aerial, let's say, plankton of insects that they could sample with this kind of traps, suction traps. The insect flies and is sucked by an aspirator in a certain height. Yeah, apart of that, and you could also try to collect insects by using a car with a collecting net, and then you could make different routes, and by this you detect maybe insect movement in places where you don't expect to be this kind of insects. But I don't know any other really study on this sense. I don't know if you.
There are a few studies. Like Miguel said, I think the trapping at the high altitudes is a very good indication. Sometimes traps like the suction traps or yellow stick traps placed in the high places is an indication, because the wind speed increases above ground as you go up. So when the wind speed is higher than the flight speed, usually it's expected these vectors are being carried by the wind, so it's a good indicator. Another evidence is sometimes the sudden appearance of adults, for example. With Acrogonia in citrus, for example, sometimes you don't see the nymphs developing the crop, and then suddenly you get a very high capture in the nymphs, like of adults by yellow stick traps. So average capture is like a half or one per trap, and then suddenly you get 5 or 10. So where do those bugs come from? Unless there are good hosts nearby, they're probably coming by movement, sometimes airborne. But there is not much data on that.
Has anybody looked for a trans-ovarian transmission?
Someone is willing to answer? I can. Since the transmission of Xylella is not a circulative, I mean, the bacterium is restricted to the first part of the foregut in the praecibarium, and there is no colonization of the insect body, I think it's wasted time to look for transovarial transmission, because there is no possibility of contamination of ovaries, at least to my knowledge. Freytag did it, yes. Freytag did it and didn't find any evidence of it. So that's true. A very old paper from Freytag.
We got a question from a nursery in Spain which considers to move the nursery to higher altitude. Do you recommend this?
Under my experience, I mean, I think, what do you mean by high altitude? Because
now, maybe 100, 200 meters.
I mean, these insects are completely ubiquitous. I mean, they are really distributed very generally in all the environments. Maybe not in the same abundance, but I think you could easily find these insects in high altitudes, this kind of high altitudes, and northern altitudes. So temperature is not also a limiting factor, because you have very nice works conducted from Finland to any other places in really cold places in North Europe. So temperature and I guess altitude is not also a limiting factor for these kind of insects. Yeah. Maybe they limit the population, but not the presence itself of the species there, to my understanding.
I agree with Miguel, and I think the most effective way is protected nurseries, screens. In Brazil, that's the way it was found to solve this problem. All the nurseries are protected. Since the production of seedlings and rootstocks and also for the mother trees, all the material is under screenhouse.
Okay, Sandy?
I know this sounds like a big point, but I think it's fairly important. How did you determine your sample size? Is it arbitrary? Did you actually analyze some data to try to get a rough guess of what it was, or what did you do?
At this point was quite arbitrary, I have to say, yeah. Yeah, maybe for next studies we could do preliminary studies to have a results on the effect size and decide on how many samples is the right size.
Well, I would not say it's purely arbitrary. I mean, our sample size is based on what we think is the minimum number of samples we need to collect in terms of the effort we could also invest on the sampling. As I said before, this group of insects, most of them never been a problem, so there are no pheromones, no traps, nothing really specifically designed to target those insects. So we are using methods that are used in general entomology. We are collecting a lot of different bags without really having a specific method for this group of insects. So, of course, we could not be on the field collecting during one day because this is not effective in the terms of sampling effort. So I think that probably right now the sampling rate, the sampling effort rate is what is really in some way shaping our sampling methodology.
I have a very basic suggestion for that, borrowed from general entomology. I would suggest that sampling intensity should be higher in those areas where the population level is very low, like in some Mediterranean areas, while in other areas where population level is higher, sampling intensity can be lower. I mean, this is a general rule, but it's true. It's a new problem. Now we look at these insects as kind of pests, but in the past they have never been looked at as a pest, so we have no previous experiences in also in sampling and defining sampling effort for this.
Another point to consider besides sample size is location of the traps. For example, after you start knowing the ecology of these insects, or where they come from the overwintering places, you can put your traps in strategic places. For example, for the sharpshooters, we know that they come from like swamps and riparian woods. It's very important to place, put more traps nearby the borders in the interface with those other habitats.
So there is time.
Just one. Okay. In our study, we use a simple random. So all the olive orchard. Going to the field, I would say that maybe for further study, we should at least think about maybe stratified random sampling, because an olive groves, especially for my experience in Liguria, that is quite complex. The environmental condition is also on a slope. I mean, there are many different variation of environmental condition that could influence the abundance density of the vectors. So maybe to take into account these, because you have high variability that can obviously influence very much the output, the results. So think a little bit earlier on the different environmental conditions inside each sampling unit, each groves, could be useful.
So time for last questions, if any, okay?
Yes, thank you. Quick question for Professor Lopes, the experience in Brazil. Indeed, we understand that vector control is key in the strategy against Xylella, and that insecticide treatment, we understand that are quite a normal practice in Brazil, and we were wondering, on top of this control of the adult stage of the insect, if there are other practices to control the juvenile stage of the insect in Brazil. And the second question, also this more a curiosity, whether there are some studies confirming attraction by Xylella infected trees of insects. So if insects are attracted by infected trees, or they just go randomly on trees. Yeah, thank you.
Okay, concerning alternative methods, there are other possibilities. They have not been much explored, because like the growers, the citrus growers, it's easier for them like to spray insecticides. And in a situation like today, we have also the Asian citrus psyllids. They are doing a lot of sprays. But we know that eggs, the sharpshooters are very vulnerable at the egg stage. There are many egg parasitoids, mimerids, trichogrammatids that occur. But citrus is not a good host for most of the sharpshooters. That only for a few. So you don't easily see like nymphs and adults. You do find for those that colonize, but they are not very often. So I think these other strategies are very important. Could be applied in the alternative hosts in these neighboring areas. So biological control could go into these places. Fungi, also the entomopathogenic fungi can be effective. There are some data on that. Trap plants also can be used for plants that are more attractive than the crops, because these bugs come from outside, from neighboring. So they usually, if you have a trap plant established in the interface, that can be important. Then you can spray insecticide in that plant. Also, some white powders, for example, particle film technology like kaolin, also change the color of the plant. So in the borders, you can also restrict movement because of behavior orientation. Concerning effect of Xylella on attractiveness of the plant, studies that have been done, evidence so far indicates that as the disease progresses, as you start seeing symptoms and clogging of the xylem vessels, usually the plant becomes less attractive. Or the bugs land, but they don't like it. So usually on the experimental work that has been done, in the early stages of infections is when the vectors, they usually land. But when the plant is really bad, showing severe symptoms, usually they try to avoid those plants.
Okay. Thank you. Thank you, everybody. And I guess we have to be back here by 11. Am I right, according to the schedule? So still thanks to everybody.